Bismillah ar-Rahman ar-Rahim. Now we're going to talk about the A2, 8 chapters which we have in the A2 syllabus, and what are the syllabus updates for the syllabus, which is 2022 syllabus, what things have been removed and what have been added to the syllabus. So we're going to talk about the topics removed and the topics added, and we're going to discuss that in a little detail. Now, in the chapter on photosynthesis, which is chapter 13, 13.2c and 13.3b have been removed, which is one is the limiting factors and it's used to increase crop yields, that's out, and C4 plants, and then the comparison with C3 plants and the anatomy and physiology of the leaves and the spatial separation of the initial carbon fixation, dependent stage, high optimum temperature of the enzymes involved have all been removed from the syllabus. Then the chapter on homeostasis, which is chapter 14, the part which says how urine analysis is used for glucose, proteins and ketones, that has been removed, but not the one which is the dipsticks, which is used in the glucose oxidase and peroxidase for glucose in blood and urine. That of course is present, but the urine analysis story in diagnosis with reference to glucose, proteins and ketones has been removed from the syllabus. Then chapter 15, which is coordination and control. In that, the whole hormonal thing is out of the course. For FSH, LH, estrogen, progesterone, and the human menstrual cycle has been removed from the syllabus. And the biological basis of the contraceptive pill has also been removed from the syllabus. These are all what I'm talking about is things which have been removed from the syllabus. Then in chapter 16, which is inherited change, the role of meiosis in gametogenesis in humans, this has been removed, and formation of pollen grain and the embryo sac, that has been removed. Now the last one is 16.2e, which is gene mutation, substitution, deletion and such, and this has moved to the AS syllabus, and I have talked about this in the previous update which I did in the video 1 of the AS syllabus. One topic which is new is epistasis. Now epistasis is previously not in the previous syllabuses, but it's been talked about in this new book. So two different genes on different chromosomes affect the same feature. The alleles of one gene affect the expression of the other, and this is called epistasis. The allele of one gene affects the expression of the other. So in chickens, if you have big G and colored feathers, small g, then you have white feathers. But if you have big F, then it prevents the production of the colored feathers. So you can see here we have got F, F, G, G, but still they're white feathers. So big F, G, G, white feathers, big F, small g, white feathers, big F, big G, still white feathers, big F and big G, small g, big F, small f, and still you have white feathers, and big F, small g, still white feathers. Now colored feathers is when there's big G, or big G, small g, but then you have to have the small f's in it, because the big F prevents the production of colored feathers. So then we have, these are the two which are colored, which has of course the big G or the big G, small g, so it's got the homozygous GG or the heterozygous GG. This is the homozygous GG, this is the heterozygous GG. But this you have to have the small f's with it. Why the small f's? Because if you have the big F, it prevents the production of colored feathers. So this information would be given to you in the question, and you'd have to figure this out. This is a question in which we have epistasis, and this is the effect, the allele of one gene affects the expression of the other, and this is called epistasis. Then I want to talk about the topic of genetic drift and the founder effect, and this I feel has got to be a question on this coming in this exam, because this is now needed to be in a much more profoundly complicated manner and more detailed form. Genetic drift is what? Is the gradual change in allele frequencies in a small population. Say you had 100,000 before, now you only have 1,000 animals left. A gradual change in allele frequency in a small population. So original population of 100,000, now they only 1,000. So what happens to the allele frequency in this? Allele frequency means the different alleles present. In what percentage were they present? Where some alleles are lost or favored just by chance and not by natural selection. So the important thing is that it has to be a small population. and there has to be a change in a gradual change in the allele frequency in a small population. So you had, say, 100,000 years, and now because of some disease or because of there's been an earthquake and they've been separated from the original, so now there are only 1,000 left. So in a small population now, how the alleles are, some are lost and some are favored just by chance and not by natural selection. So the numbers has to decrease. Now, two terms that we must be very clear about. Number one is gene pool. What is gene pool? The complete range of DNA base sequences in all the organisms in a species or population. Like in the human beings, we have what is the entire genetic information, the DNA base sequences on our 46 pairs, on 46 chromosomes. But that plus every other human being, what are all the different alleles? Like you have 3 alleles for blood group. Similarly, for hair color, for skin color, for eye color, you would have different alleles. So the complete range of DNA base sequences in all the organisms in a species, that's called the gene pool of humans. Now, as opposed to that, we are talking now of another term which we've come across as founder effect. Again, this is a reduction in gene pool. Reduction in gene pool compared with the main populations of a species, resulting from only 2 or 3 individuals. So we've only got 2 or 3 individuals which are now left to breed, starting off a new population. Starting off a new population. So reduction in the gene pool. Founder effect is the reduction in a gene pool compared with the main populations of species, resulting from only 2 or 3 individuals who only have a few of the alleles, starting off a new population. Then another term which I want to be talking about is evolutionary bottleneck. Now, evolutionary bottleneck is what? It's a time period when the number of a species falls to a very low level, resulting in the loss of a large number of alleles and therefore a reduction in the gene pool of the species. Please remember, founder effect was only a few individuals. That was a separate story. Here it is an evolutionary bottleneck is a period when the number of a species falls. So again from say 100,000, now we are left with only 50. And this, of course, these 50 have a couple of alleles. They don't have all the alleles which are present in the 100,000 animals. Therefore, reduction in the gene pool of the species. So now these 50 do not contain all the alleles. So now there is a reduction in the gene pool, and this is called evolutionary bottleneck. Now in sympatric speciation, you see we are doing 2 speciations. One is allopatric, in which they get separated geographical barrier. But in sympatric speciation, we don't talk about the previous things that we used to talk about in an old syllabus. Now we talk of 2 things, ecological separation and behavioral separation. So ecological separation is what? It's the separation of 2 populations because they live in different environments in the same area and so cannot breed together. One lives in a desert, one lives in some forest, and so they are just separated ecologically, live in different environments in the same area, same habitat, same area, but then they lived in a different environment, so they cannot breed together. The other is behavioral separation. In behavioral separation, the separation of 2 populations because they have different behaviors which prevent them breeding together. Maybe they are in the same pond, maybe they are in the same river or in the same ocean or in the same lake, but because they have different behaviors, so it prevents them from breeding together. So this is called the 2 sympatric speciations now that we have to teach you is ecological separation, different environments. Ecological separation is different environments, E for ecology, E for environment, and behavioral separation is because they are in the same place, because they have different behaviors which prevent them from breeding together. Then some new words have been used in the definition of species. The first one which we use usually is the biological species. A group of organisms with similar morphology and physiology which can breed together to produce fertile offspring and are reproductively isolated from other species. But the one which is morphological species is a group of organs that share physical features that distinguish them from other species. So that's only morphology, the physical features that we are talking about. Then ecological species, a population of individuals of the same species living in the same area at the same time. So these would be then called ecological species. So one area, maybe they're living in a desert in the Sahara and Sahara Desert. So the population of individuals of the same species. So these will be called ecological species. Then the population of all of the organisms of the same species present in the same place and at the same time that can interbreed with one another. So there are four different names that they have given you now: biological species, morphological species, ecological species, and of course the definition population. If, like we say, the population of Pakistan is whatever number it is. So it's the number of human beings, whether they are old, whether they're young, whether they're sick, whether they're healthy, whether they're children, whether they're male or female, is all of the organisms of the same species present in the same place. So the very nice example is population of Pakistan is the number of people who live in Pakistan. Modification in the word defined bioinformatics has been changed to how the information is handled. So even though bioinformatics is now not named in the syllabus, but still we need to know about it. So what is bioinformatics? The collection, processing, and analysis of biological information and data using computer software. So basically, it's the age of the computers, and all your students who decide to specialize in computers, that's the thing which is going to be the main source of income for the futures. Now, DNA sequences is a fully automated process, and the base sequences of whole genomes and many individual genes are known now. Now, what do we do with all this information? Well, all this data on the DNA and protein are stored in the digital form in a variety of databases. So the DNA tells us the sequence of bases, the proteins tells us the sequence of amino acids. So we want to study pepsin from a cow, then we want to study pepsin from a monkey, and then we want to study pepsin from a human being. So we compare the structure of pepsin or the genetic information of pepsin, how the amino acid sequences vary. So all this information is of course saved on the computers, and we have all these huge databases. Then we can do comparisons, can be made with other known genomes. For example, the human genome can be compared to the genomes of the fruit fly, Drosophila, or the nematode worm, or the malarial parasite Plasmodium. So sequences can be matched and degree of similarity calculated. Similarly, comparison can be made between amino acid sequences of proteins or structures of proteins. Close similarities indicate a recent common ancestry. This is the important thing which you need to remember. Close similarities indicate recent common ancestors. Then the human genes can be concerned with the development can be found out, and we can study how do organisms develop, what happens, which genes are switched on, which genes are switched off. So microarrays can be used to find out when and where genes are expressed during the development of an organism. And of course, this research helps us to find out how we can maybe avoid certain diseases, or we can how this development can be then affected if something is added to it, and how we can change this development, or at least maybe we can help in the treatment of certain diseases if we know that if this didn't happen. So all these information is very interesting to help us become this world to become a better place. So bioinformatics, this is the you have to understand what is bioinformatics, not just the name, not just the define, but the way how we handle this and what this information means. Here's another topic which is now newly been assessed or nearly been written in the new book, and I want to be talking about those topics as well because I feel there's something they've been added to the book for a certain reason. Now, this is actually a treatment is the procedure for carrying out gene therapy is in SCID. Now, SCID is a disease in which the immune system of a child, when born with it, it cannot function. So what we do is we get the correct sequence of the ADA, the enzyme which is not working in people, in children with SCID. So we get that, we get the DNA with the correct base sequence for ADA, and then we get a virus, and then we insert the normal allele of the ADA into the virus. Now we've got this. Now this is recombinant DNA. And then what we do is we take out stem cells
we take out stem cells. Now these are the stem cells. We've taken out stem cells from the bone marrow. You can see here we're doing it here from here to here. We've taken out the stem cells. So the stem cells, and then we have what we do with the virus insert. So we've added the virus to it, and the virus inserts the normal allele of ADA into the stem cells. And then what we've done is the stem cells with the normal allele of ADA is injected back into the blood of the child. So this has gone back into the child. Now the stem cells will divide and differentiate into T lymphocytes, and of course they have the functioning ADA, so then the child's immune system will develop. Please remember, these children without an immune system were enclosed in plastic bubbles. They had no chance of survival. But now with gene therapy, they can have some chance of survival. And maybe as years go by, some better treatments will come out. But this is the latest that we have on gene therapy and how gene therapy can help children with SCID. So first taking out the stem cells, because you see, if you took out the lymphocytes and you changed them and added the normal thing, but you need to give regular transfusions of that. But here, because now we are putting the gene of choice into the stem cells keep on dividing, and they, of course, will not to be replaced. So we don't need to give regular infusions or transfusions into the child. So we must think of things in which we do things which is more permanent rather than temporary solutions to a problem. So this is something new which I needed to talk about. Now, a little portion of this is new in the syllabus, sort of new. Is the need for energy in living organisms. Why do we need energy in living organisms? And the need energy is basically we need energy, and the ATP is the universal energy currency. So the number one point, which is for active transport, moving anything, substance must be moved across membranes against their concentration gradients by active transport. We study a lot of that in the kidney. Then the movement of organisms. Movement of organisms is, of course, we can talk about muscle contractions so that we can move from one place to the other. Or we can also talk of movement is when something is made on the ribosomes and then it is transported to the Golgi. And then, of course, the Golgi is going to modify it, and then a Golgi vesicle is going to sort of then be formed, and then it's going to move out of the cell. Then the third one is anabolic reactions. Anabolic reactions, the word has been used here, anabolic reactions. So anabolic reactions, it means monomer to polymer. So monomer to polymer means nucleotides to DNA. So that happens in DNA replication. And then protein synthesis, which is amino acids to proteins. So any of these, either we talked of anabolic reactions is DNA replication or amino acids to protein. We can also talk of other anabolic reactions like glucose to glycogen in human beings when it's stored in the liver, or we can talk of glucose converted to starch in plants when photosynthesis occurs. So you have a lot of things from which monomer to polymer is an energy requiring process. So the need for energy in living organisms.
Now, the next thing new in the syllabus is this part of the syllabus which is marked with a line and this part with a line. So it is the protons returned to the stroma from the thylakoid spice by facilitated diffusion through ATP synthase, providing energy for ATP synthesis, and details of ATP synthase are not expected. And here it is GP to TP in reactions involving reduced NA. So this is, wherever there is this sort of a line here, a bar line here, that indicates that these things are new in the syllabus. Some things, of course, I've been mentioning are in the book, but they're not in the syllabus very clearly mentioned, but I've talked about that as well. Now, a quick recap of what we did in the photolysis and all that. So, photophosphorylation is the ATP which is made by the light story. So, electrons are excited in photosystem 2, and then electrons are excited in photosystem 1, and they're accepted by the primary acceptor. And then, when these electrons fall back, they pass through these carrier cyclic electron flow and then the cytochrome complex, and then energy for chemiosmoic and synthesis of ATP. And this ATP is made, and this is called non-cyclic photophosphorylation. And the other one, the electron goes on to the NADP reductase and NADPH is made. So ATP is made and NADPH is made. Then the cyclic electron flow, we have to be very clear about. It's only PS1 which is involved, and the electrons excited by light, they come back. They don't go into the NADP reductase story. They don't go this side. They don't go this side, so they will not go this side, and they will return back, and then they will go through the electron transport chain and ATP will be made. So this formation of ATP, which is only involving photosystem 1, is going to be called the cyclic electron flow. So this is going to be the cyclic electron flow. The reason being that we require 9 ATPs and we require only 6 NADPH. So this story, the non-cyclic is going to go 6 times. So we're going to have 6 ADP and 6 NADPH. But then we've got to get the additional 3. We need the cyclic to go 3 times so that we get the 9 ATPs. Now just an overview of ATP synthesis. Number 1, electrons are harvested and carried to the transport system, number 1. Then number 2, electrons provide energy to pump protons across the membrane. And then of course you see here what has happened is that there's pyruvate from the cytoplasm, acetyl-CoA, Krebs cycle. And then of course the 3 oxygen joins with protons to form water. We've got that here. And then we have protons diffused back in driving the synthesis of ATP. So protons will diffuse back in through this channel protein which is called ATP synthase, and just a recap of this, and then we're going to get the ATP. So this is called oxidative phosphorylation. Just please pause the video here, have a look at it, go through it very point wise. Now if you look at the story as compared to respiration, let's compare this with photosynthesis. Now let's have a quick look at this. What are we talking about? Now everybody please be very careful. This is the stroma. This is the stroma. This is the thylakoid membrane. This one is the thylakoid membrane. And inside here we have the thylakoid space. But in photosynthesis what is happening? How is ATP being made? You see the hydrogen ions. The hydrogen ions are here outside in the stroma. They are being moved into the thylakoid space. So you have a lot of hydrogen here in the thylakoid space. And then all this hydrogen, this one here, all the hydrogen has to pass through the ATP synthase. And that is when ATP is made. And this ATP is going to go into the Calvin cycle, which is going to take place in the stroma of the chloroplast. So look at this situation as compared to this. Now this has been specifically mentioned in this syllabus. Where have we taken this diagram from? This taken diagram from here. What are we talking about? We're talking about the light dependent reaction. We're talking about how the electron transport chain works on the thylakoid membrane. And I'm going to do a comparison with the mitochondria so that you can then really follow this very clearly. So hydrogen ions moving from the stroma into the thylakoid space. Then the hydrogen ions will, as they cannot cross the membrane is a phospholipid bilayer, so ions cannot cross this. So they have to pass through one ion channel, and that is the ATP synthase molecule. And when they flow through that, then there's, the catalysis is going to take place. There's an enzyme which rotates, and ADP and inorganic phosphate combine to form ATP. And this, of course, is called photophosphorylation because it happens in the presence of light. Here you can see photosystem 2, here you can see photosystem 1, here you can see the NADP reductase. You have to be very clear about this. What am I talking about? I'm talking about the thylakoids in the chloroplast. So the thylakoid membrane, this is the thylakoid membrane, this is the thylakoid membrane, this is the thylakoid space. Now this is a very, very nice comparison between the chemiosmosis in mitochondria and chloroplast. And you can see the color, the key, higher hydrogen, lower hydrogen. But the important thing to you to see is this is the matrix here, this is the stroma here. This is the thylakoid membrane, this is the inner membrane. And this is the intermembrane space, and this is the thylakoid space. A very nice comparison, and I bet there's going to be a question on this. So it's a comparison. Now the comparison is that in that situation the hydrogen ions were being pumped into the intermembrane space, and then they flew to, then they passed through the ATP synthase. Here they are entering the thylakoid, so from the stroma they are entering into the thylakoid space, and then the ATP synthase is on the thylakoid membrane, while the ATP synthase is on the inner membrane. Here the ATP is on the ATP synthase is on the thylakoid membrane. Now comparison is very nice. Mitochondria, E source is an organic molecule, the electron source is an organic molecule. In chloroplast, the electron source is water. Mitochondrial matrix is analogous to the chloroplast stroma. Proton gradient in mitochondria is established in the intermembrane space. In the thylakoids is in the stroma. So from the stroma they are flowing into this thylakoid space. However, ATP is generated on the inside of the mitochondria matrix but the outside of the thylakoid. So ATP is made on the outside of the thylakoid, while the ATP is on the inside, is generated on the inside of the mitochondria, inside the matrix. So this is a very, very nice comparison. Please pause and have a look at it and then go through it. And this is of course something which is new in the syllabus, and we need to talk about this. A quick recap of the Calvin cycle. Now, the Calvin cycle is also called the light independent stage. I do not agree with that because it is not light, it's not sort of directly dependent on light, but it is dependent on the products of the light dependent stage. So, when you say light independent, and I can think of a suggest question which you could get on this, in which they say, okay, why is it wrong to call it the light independent stage?
Slight change in the syllabus is not the change in the syllabus is, but you've got to know what is GP and what is TP. GP is glyceraldehyde phosphate and TP is triose phosphate. So this is the name that we give in A levels. But then the important thing is the ATP, which is used to, of course, activate it, and the NADPH, which is going to the H is going to reduce it. So this is the phase 2, which is the reduction, which is this phase. They've called this phase reduction phase. And then, of course, this phase is the carbon fixation phase. And then this phase is the regeneration. So you can see the three different colors which have been used to depict this. Now, basically, what they want you to know is the reduction part. That the reduction is the ATP, of course, is not going to reduce it, but NADPH is going to be reducing the GP to TP. So GP is reduced to TP because NADPH gives it the hydrogen. So NADP is regenerated, and the reduced NAD is now no longer than just NADP. So this is the point of the syllabus which you must be knowing. Then coming to this part, the Calvin cycle, part 2 reduction. For every 3 carbon dioxide molecules, 6 G3P are produced, but only 1 is the net gain. 3 RuBP, 15 carbons in total. So 6 GP contain 18 carbons, a net gain of 3 carbon. 1 GP exits the cycle, the other 5 are recycled to regenerate 3 RuBP. So these, of course, these numbers are being churned out because otherwise we can't tell you how a 3 carbon exits. So how a 3 carbon exits is, unless you come up with these points, is that there are 15 molecules of this, then there are 3 of these, so 18, and then we have a 6 carbon intermediate which is forms here somewhere, and then we have a 3 carbon. So still 15 and 3 is 18, and then this is also 18, and then 3 exit, and then we are left with 15. And then these 15 are regeneration of the CO2 acceptor, which is the RuBP, which has to be regenerated. That completes this video, and I will continue. There's some points still left, which I will continue in a second video very soon. Thank you very much for watching, and please do subscribe and do leave your comments if I can help you in any other way. Thank you very much.